Hi, my name is Clarice Risseeuw, and together with Groutars, I'll happily present our paper entitled Flavorium, an exploration of Flavobacteria's living aesthetics for living color interfaces. Our co-authors are Colin Ingham, Hamidjaja, Willemijn Elkhuizen, Sylvia Pont and Elvin Karana. For Material Experience Lab at Delft University of Technology, Material Incubator Lab at Advanced University of Applied Sciences in collaboration with biotechnology company Humidity. Our work contributes to the ongoing work in biological HCI by exploring the potential of Flavobacteria as responsive medium. Here we envision interfaces that communicate to us through vivid living color, created by these fascinating organisms. Flavobacteria are marine bacteria, which can for example be found in coastal waters around the globe. These non-harmful bacteria produce optical structures that interact with light, resulting in very vivid angle-dependent color. Such structural color can also be found in the feathers of a peacock. Microbiologists have researched Flavobacteria's ability to self-organize in relation to various environmental factors. However, despite their vivid colorations and highly responsive behavior, Flavobacteria are yet to be explored as a living medium for interaction design.
From an interaction design perspective, our interest in these microorganisms lies in the fact that their colonies change in relation to various environmental factors. This gives us a lot of room to play and influence the way in which their appearance changes over time and the experience that this brings about. Here our focus lies in this organism's living aesthetics. Living aesthetics is a term that relates to the way in which we as humans experience the type, degree and duration of change inherent to the growth, reproduction and decay of an organism. Designing with these living organisms does require us to properly understand this temporal aspect in order to purposefully design with it. So here we see how a colony of Flavobacteria typically develops over the course of days. We can see the edges of the colony expanding outwards. These outer edges contain the youngest bacterial cells, which are typically more vivid and multicolored in their appearance. Once these cells grow older, as can be seen in the middle parts of the colony, they also tend to lose their iridescent color. Based on this, we were able to quantify this changing appearance of Flavobacteria by means of their form, texture and iridescent color. Form describes the overall shape of the colony and how it expands over time. Texture is determined by the distribution of iridescent sections across the colony, as not all parts are iridescent at the same time. Thirdly, the iridescent color will also change in hue and brightness over time and across the surface of the colony. So this begs the question, how can we steer their living aesthetics by means of an input mechanism? As mentioned, these bacteria have proven sensitive to numerous environmental factors, and here we zoomed in on the humidity present in their environment as a suitable input mechanism. This because the humidity directly influences the way in which they move and form their optical structures. So we then proceeded to investigate how humidity influences these bacteria, their growth and corresponding living aesthetics.
But how can we capture and characterize Flavobacteria's living aesthetics? This was the first challenge we faced in our research. We developed a capture tool which automatically captures a petri dish while varying the viewing and illumination angle. As can be seen in these binary images, bacteria colonies grown in high humidity tend to expand faster and result in amorphous shapes, while bacteria grown in low humidity result in a circular shape that appears hollow due to the fine textures of the colored parts. All the colonies appeared the most brilliant and multicolored from a viewing angle of 45 degrees. Looking across the different levels of humidity, the high humidity samples appeared slightly more multicolored. During the study, samples were stored in an incubator and only captured every 2 days, leaving gaps of information on how the living aesthetics developed in between. Additionally, the samples grown at high humidity reached the limit of their habitat in a matter of 5 days, making it impossible for us to study this behavior over longer periods of time. To overcome these drawbacks, in exploring the potential of Flavobacteria for living color interfaces, we developed Flavorium.
Flavorium is a custom-made habitat that allows for large-scale growth of Flavobacteria. It also allows for digital control over the relative humidity in the habitat and frequent automated documentation of their growth. During our initial test runs with Flavorium, we were able to observe the behavior of Flavobacteria over the course of a month. With constant humidity levels, we could already observe differences in expansion rate and texture across the colony. In the future, we also aim to introduce changes in growing circumstances such as the humidity and see how Flavobacteria respond. The development of this biodigital research artifact thus enables us to envision and test future HCI applications that involve these living organisms. Inspired by the existing body of work within the bio-HCI community, we envisioned 3 categories in which Flavobacteria as a living medium can either embody digital data, communicate environmental signals, and allow for traces of skill and use. For example, we envision the concept of a living monitor, in which Flavobacteria's growth is digitally controlled and used to represent abstract digital data in a tangible manner. In another example, Flavobacteria can also directly sense changes in their environment and communicate these through their living aesthetics. In the concept of a living label, Flavobacteria communicate and amplify the well-being or struggle of a houseplant by reacting to the humidity of the soil. Thirdly, the peculiar ways in which humans interact with objects and spaces can be captured through Flavobacteria's living aesthetics, enabling a patina of living traces to grow on things of daily use. A playful take on this is our concept of living notes, where Flavobacteria can be used to create temporal messages. So, in conclusion, our work presents a design space for Flavobacteria as a living medium for living color interfaces. We provide a method for quantifying their living aesthetics in relation to a specific input mechanism, and from this we developed Flavorium to enable long-term study of this organism's behavior as well as a means to control it. And with this we provided the first steps towards understanding the dynamic and complex nature of this living organism from an HCI perspective. We believe that in this complexity, as challenging as it is, there are also many opportunities for discovering novel ways in which humans can interact with living organisms. Thank you very much.